So now we're going to look at some traits and some disorders that are inherited differently than everything we've seen so far. So all of the traits that we've looked at so far are called autosomal. That means they're found somewhere for humans on the first 22 chromosomes, but basically the non-sex chromosomes. The last pair in humans, the 23rd pair, are called the sex chromosomes. And as you can see here, they're different in males and females. Males have an X and a Y, and you can see they're non-homologous, whereas females have two X's. And so when we talk about X-linked traits, we're going to talk about genes that are found on the X. And notice that since females have two copies and males only have one, that's going to change how they're inherited. So the first thing you need to know is that females are XX, males are XY. And going back to our Punnett squares and meiosis, notice that it's the mother who can only give the X. So half of her eggs are going to get one X, half of her eggs are going to get the other X. And it's the father who's going to be determining the sex of the baby. So it depends on which sperm fertilizes the egg. Half of the sperm have an X in it, half of it have a Y. So if it happens to be fertilized by a sperm with a Y, then we have a male that's formed. If it happens to be fertilized with an sperm that has an X in it, then it's a female. There's always a 50-50 chance of having a male and female. So as we go through the traits in sex-linked traits or X-linked traits, notice that we always need to put male or female in addition to the trait that we're talking about. A scientist by the name of Thomas Hunt Morgan studied fruit flies. Their scientific name is Drosophila melanogaster. And he realized that eye color in fruit flies was not inherited equally between the male and female fruit flies. And so he hypothesized that the gene must be on the X chromosome and that there was no corresponding gene on the Y chromosome. Why did he work with fruit flies? Mainly because they had a very short life cycle. They were very easy to take care of, and he could have a lot of offspring, which meant a lot of data in a short amount of time. In humans, there are two traits or disorders that we're going to be looking at. Color blindness, and it doesn't really mean they can't see any color. It usually means they have a hard time distinguishing between different shades or just certain colors. Hemophilia is another X-linked trait, and that is where your blood is missing some proteins that help it clot properly. So whether people have a cut, which is where you're bleeding on the outside, or a bruise where you're bleeding on the inside, it doesn't clot, and so that can cause problems. Because it's an X-linked trait, we're going to show these as superscripts that are only on the X chromosome. And the ones we're going to be looking at are all going to be sex-linked recessive. So the disease or the trait is going to be the little letter, and the capital letter is going to be normal. So notice in females, you have three options. The female can have X capital H, which is normal, X capital H, X lowercase h, which is a carrier, and X little h, which is a female with hemophilia. Or in this case, just notice that this is homozygous recessive, this is heterozygous, and this is homozygous dominant. Now, the reason these females are not called normal, even though they look normal, it's because they're carriers. They can still pass the diseased allele or the allele with the trait to their offspring. Now, notice with males, we don't have three options. We only have two. Males cannot be carriers because there's nothing on the Y chromosome. So the gene, the X-linked trait or disorder is only going to be attached to the X chromosome. So they're either normal or they have the trait or the disorder. The reason that these are a lot more frequent in men is because they only need one of the lowercase letters on their X chromosome in order to have the disease or trait. Females have to have two in order to have the trait or the disease.